Hello everyone, we are honored to share our research genomic epidemiology and lineage-specific risk stratification of TET, X4, mediated tigecycline resistance along the pork production chain, a One Health perspective. Antimicrobial resistance is rising worldwide. Tigecycline is a last resort option for MDR gram-negative bacteria infections, but its efficacy is increasingly compromised by rapid dissemination of plasmid-borne TET, X4. However, high-resolution and farm-to-table genomic data targeting TET, X4, positive ice along the pork production chain remain limited. Surveillance identifies slaughterhouses as key enrichment nodes in the spread of TET, X4, positive E. coli along the pork chain. Lineage risk stratification reveals hybrid high-risk E. coli clones. Occupational exposure poses higher risks than general community contact. Conserved plasmid backbones drive TET, X4, persist in food systems. Overall recovery rate under tigecycline-based selective enrichment was 58.12% across the pork chain. The isolate population was dominated by E. coli, and TET, X4, was the primary resistance gene. Farms and slaughterhouses showed the highest recovery rates of TET, X4, positive E. coli, X4, positive E. coli rate in pig farm workers was markedly higher than in the general population. AST showed an extremely high MDR rate, 99.9%. A total of 212 ARGs were identified. Farm isolates had the broadest resistom, while human isolates showed enrichment of PMQR and selected beta-lactamase families. A total of 730 virulence genes were detected. XPEC was the most. UPEC and EPEC were present at low frequencies. NMEC was limited to non-human sources. STEC and ETC were almost absent. TET, X4, positive isolates were dominated by extraintestinal rather than classical diarrheogenic pathotype. WGS analysis of 790 TET, X4, positive E. coli showed a population dominated by phylogroups A and B1. ST10 and ST195 were the main prevalent lineages and were widely detected in farms, slaughterhouses, pork, and wastewater. Using a less than or equal to 10 core genome S cutoff, 2,574 putative clonal transmission events were identified, with more between-source than within-source events. Clonal links were concentrated at the farm-slaughterhouse retail interfaces, marking these nodes as key control points for TET, X4, positive E. coli dissemination. Integrating data from NCBI, we constructed a global database of 1945 TET, X4, positive E. coli isolates for lineage-level risk stratification analysis. Based on ARG and VF burdens, isolates were classified as low/baseline, high VF only, high res only, and hybrid. Hybrid isolates accounted for 26.7%. Hybrid isolates showed convergence of resistance and virulence. ST195 was resistance dominant, ST48 was virulence enriched, and hybrid isolates spanned multiple ST backgrounds rather than a single epidemic clone. A total of 234 TET, X4, carrying plasmids were retrieved from NCBI, covering 47 replicant types, with INC-F/INC-HI/INC-X1 as the dominant backbones. E. coli was the key host for TET, X4, positive plasmids, with pig-derived and China-origin plasmids account large proportions. TET, X4, positive plasmids frequently co-carried multiple ARGs, including last-resort resistance determinants such as MCR and TMEC-CD3 TOPRJ3. A conserved flexible genetic structure centered on RDMC TET, X4, and mediated by ISBSA3/IS1/IS26/IS15, together with TA system double lock maintenance, may promote plasmid stability and horizontal transfer. This study systematically mapped TET, X4, mediated tigecycline-resistant E. coli across pork production chain in China from a One Health perspective. Farms and slaughterhouses were key dissemination and enrichment nodes. Lineage-specific risk stratification identified two routes: resistance-heavy commensals in processing nodes and resistance convergence in hybrid lineages. TET, X4, positive plasmids were driven by INC-F/INC-HI/INC-X1 backbones, and maintained stable transmission through the conserved RDMC TET, X4, genetic module and TA system.